Okay. Hello everyone. Today I will share my work about the catalogue of small proteins from the global microbiome. Small ORFs are small open reading frames shorter than 100 codons. Recently, a series studies have shown that small ORFs and small proteins they encode can perform crucial roles in microbial cells, such as they can stabilize the large protein complexes, regulate the transporters. Also, they can perform the antibacterial activities, or they can perform the signal transduction pathways. However, to identify small proteins, uh, there are still a lot of limitations in both computational and experimental sides. First, when annotating genes from genomes, the traditional gene annotation methods often use the 100 codons as the cutoff to prevent from false positives, so the existing algorithms are not ideal for annotating small ORFs. In addition, the small proteins, because they are too small, so they can easily escape from the biochemical enrichment, and they are very hard to be detected by mass spectrometry because of the incomplete small protein databases for now. So small proteins have been ignored due to the limitations for a long time. Recently, with the advance of technology such as RiboSeq, a lot of functional small proteins have been discovered. In 2019, Sparrow reviewed over 4,000 conserved small protein families in human microbiomes and further investigate their potential functions. However, for small proteins in different habitats, our understanding is still very limited. So here we want to construct the global microbial small ORFs catalog called GMSC. In general, we predicted small ORFs from over 87,000 microbial genomes from Progenomes 2 database and 63,000 publicly available assembled metagenomes from the global hepatides from a SPARE database. And then after removing redundancy, we clustered the small f's into families at 90% amino acid identity. The sequences in our catalog range from 9 amino acids to 100 amino acids, and our samples are mainly from human gut, soil, and quantic. Then to facilitate the use of our resource, we developed a tool called GMSC Mapper to identify and annotate small proteins from microbial genomes or metagenomes. So the users can provide their own genomes or directly the gene sequences. And after prediction and homology searching, they can obtain the full annotations of small proteins, such as taxonomy, habitat, and quality. And now the tool can be easily downloaded by Bioconda. And now the resource is publicly available in the GMSC website, so the users can query small protein sequences by GMSC mapper integrated in the website, or they can search their own interesting small proteins by hepatides or taxonomy. And here is the website. Then we want to further investigate the functions of these small proteins. So we assigned a concept domain for these small proteins using CDD database, and we found that the vast majority of multigenous small protein families have no detected concept domains, and many are also annotated with domains with unknown functions. For example, for this small protein family, they present in multiple phylum and occur in all the 8 major hepatids, but it still cannot be annotated with any conserved domain, so their function is still unknown. So we found that even the conserved small proteins still lack functional annotations. And for the annotations ones, they are mostly annotated with the ribosomal proteins, or they belong to the helix-turn-helix P5 clan, which is associated with DNA binding function. Then within the whole small protein pool, we focus on specific functional peptides, antimicrobial peptides. This work is led by Celu. In general, directly from our small F's catalog, we use machine learning to predict about 1 million candidate antimicrobial peptides, and then we tested 100 synthesized peptides in vitro, and 79 of them shows the antimicrobial activities. Furthermore, we conducted the in vivo verification of them, and some of them shows the anti-infective activities in the mouse model. And now the results of antimicrobial peptides is also publicly available in the MPSphere website. In summary, we constructed the global microbial small apps catalog from the isolated high-quality microbial genomes and publicly available metagenomes across global distinct hepatides. And we now the resource is publicly available in the website to download and browse. And here is the QR code for the website. And to facilitate the use of our resource, we developed a GMSC mapper for the users to search their own small proteins against our catalog. For the large range of small proteins, we investigate their functions, but we found that even the conserved ones, most of their functions are still unknown. So we focus on antimicropeptides and tested and validated some of them. In addition, we also conducted a lot of ecological patterns of small proteins, and we found that bacteria harbor more small proteins proportionally than archaea. And also we conducted a series of quality tests for our catalog in both computational and experimental methods. If you are interested in the further results, please go to the preprint to see the details. In our catalog shows an immense and explored diversity of small proteins, and it can be used to study the presence, prevalence, distribution, and potential ecological roles of small proteins on the global scale. If you are interested in this work, please feel free to contact me. Finally, I will thank my Big Data Biology Lab led by Louis for their kind support, and I would like to thank all the co-authors in this work. Thank you for all. If you have any questions or suggestions, I will be very welcoming.